So welcome back again. We're picking up where we left off after the last recording. And remember, I stopped at the concept check. So here's the concept check again. Which stage or stages of cellular respiration occur in the cytoplasm and which occur in the mitochondrion? And how do redox reactions, and I probably worded this poorly, I should have said, how do redox reactions make free energy available from molecules? Okay, so what stages of cell respiration occur in the cytoplasm? Only glycolysis. It occurs uniquely in the cytoplasm. And what stages occur in the mitochondrion? The citric acid cycle and that two-step process called oxidative phosphorylation, which involves the action of the electron transport chain and chemiosmosis. So hopefully you got those. How do redox reactions make free energy available from molecules? By moving electrons from non-polar covalent bonds to polar covalent bonds, from a low electronegativity, high potential energy situation to a high electronegativity, low potential energy situation. Moving from non-polar covalent bonds, which is high potential energy, low electronegativity, to polar covalent bonds, high electronegativity, low potential energy. Those are actually very important connections to make. There is this inverse relationship between electronegativity and free energy or potential energy. So if you have high electronegativity, you have low free energy or low potential energy. If you have low electronegativity, then you have high free energy or high potential energy. And that's going to be a very important connection to bear in mind when we take a look at the electron transport chain, which we will do in this section. So onwards and upwards. So the first step in cell respiration is glycolysis. Now, there are a number of diagrams in the textbook, and I've got them here in the presentation and in the slides that you can download, that show the chemical reactions. The good news for you is I don't care whether you learn the chemical reactions or not. In fact, if you don't want to learn them, don't, because I'm not really that interested in you knowing the sequence of chemical reactions at this point. What I really want you to know is this: what chemically goes into glycolysis, what chemically comes out of glycolysis, energetically what goes into glycolysis, energetically what comes out of glycolysis. Those are the things that I want you to really know. What are the chemical reactants? What are the chemical products? That's what goes in and what comes out chemically. Energetically, what do you have to invest in the process of glycolysis? Energetically, what do you get out as an end result of glycolysis? That's what I want you to focus on. So will I show you the diagrams with the chemical reactions? Yes. Do I expect you to know them? No. So we can break the chemical reactions down into two basic steps. One is referred to typically as the energy investment stage. The other is referred to as the energy payoff stage. And the reason why that is so is because of the nature of glycolysis and the chemical reactions that are involved. Remember, we talked about the idea of the activation energy for the breakdown of a molecule or for a chemical reaction, the amount of energy that you have to put in to destabilize a molecule so it will participate in the chemical reaction, the activation energy. Turns out that glucose has a very, very high activation energy. So yes, the chemical bonds of glucose, those non-polar covalent bonds, represent a very good source of chemical potential energy. And a significant amount of free energy can be released from breaking those non-polar covalent bonds. But it's also in a fairly stable state. It requires an investment of energy to destabilize it. So much so that you actually have to invest 2 ATP molecules per glucose molecule at the beginning of glycolysis. So this is why this is referred to as the energy investment stage. So here's what the chemical reactions of the energy investment stage actually look like. And you can see the investment of the ATP involves two separate steps. And it turns out that the key step here is this last step, where the second ATP is actually invested. Because before we're done with looking at cell respiration, we're going to talk about how cell respiration can be regulated. And it's this step where we invest the second ATP that we carry out the first irreversible step of cell respiration. So this is where cell respiration is actually regulated via this step. There'll be more to talk about here when we get to the end of the presentation, the end of the chapter. So this is where we invest the ATP. The net product of this is two molecules, one called G3P, which you're going to encounter again in photosynthesis, and another one called dihydroxyacetone phosphate, or DHAP. And you've encountered both of these before when we were talking about carbohydrates back in chapter 5, because these were actually the 2 3-carbon sugars in the table of monosaccharides. And it turns out that these are structural isomers of each other. The problem for cells is that only G3P, glyceraldehyde 3-phosphate, can be further processed to pyruvate to start the energy production associated with glycolysis and cellular respiration. So if we did nothing with the DHAP, we would throw away half the energy of a glucose molecule. And as we have seen multiple times and will continue to see for the rest of the semester, cells never waste anything. Here's energy to be obtained. So there is an enzyme called isomerase that carries out the conversion of DHAP to G3P. So as a product of the breakdown of glucose, we don't get one G3P, we wind up getting 2 because of this conversion of DHAP to G3P. So as we follow along here in the energy releasing part of glycolysis, the energy payoff, we need to remember that while we are only showing a single G3P and a single molecule throughout all of this, we need to remember that we're doubling down on all of this, because the DHAP is converted to a second G3P that is also processed. And that's the reason why it's not one NAD plus, but 2 NAD pluses that are required in this step. And in this step, some electrons are released that can be used to reduce NAD plus to NADH. So we get 2 NADHs. Here's a capture of some electrons as energy. That then allows us, through substrate level phosphorylation, as we do this conversion, this chemical conversion, to generate 2 ATPs. So we've got back the 2 ATPs that we invested. So now we're all square as far as ATP is concerned. But as we follow along, we recognize that in the last step of glycolysis, we generate 2 more ATPs, again, through substrate level phosphorylation. The phosphate on the molecule PEP is transferred to ATP to give us pyruvate. And again, remember, there are 2 G3Ps, 2 of these, 2 of these, 2 of these, 2 of these, giving rise to 2 pyruvates. Oops. What I want to do here. So in this diagram, we can see all of the chemical reactions. We can see where the NADHs come from. We can see where the ATPs come from. Notice that I've said here that the net yield is 2 ATPs. That takes into account the fact that, yes, while we get 4 ATPs out, we had to put 2 ATPs in the first place. So we put in 2, we got back 4. So our net yield, or our overall yield, is 2 ATPs. We also get 2 NADHs out, and we get 2 pyruvates out. So if we want to summarize all of this in terms of reactants in, products out, energy in, energy out. This is how we would summarize it. So the one glucose molecule goes in chemically. What comes out is 2 pyruvates. Energetically, what goes in is 2 ATPs, but energetically, what comes out is 4 ATPs plus 2 NADHs. Now, you could argue that the electrons in these NADHs are in an unusable position because they're out in the cytoplasm, and as you'll see, the electrons in NADH are utilized inside the mitochondrion. But the good news here is those electrons can be transferred into the mitochondrion, not the NADHs, but the electrons themselves. There is a way of getting those electrons into the mitochondria. So these are not lost. All is not lost here. These electrons can and do wind up in the mitochondria and contribute to the generation of ATP. And we actually take that into account in our energy budgeting. So if we're going to summarize the whole thing, chemically, glucose goes in, 2 pyruvates come out. Energetically, 2 ATPs go in, 4 ATPs come out, net yield is 2 ATPs. 2 NAD pluses go in, 2 NADHs come out. Remember, the electrons in those NADHs are usable, as we'll see. So this is the diagram to focus on as far as glycolysis is concerned. Don't worry about the chemical reactions, worry about what goes in, what comes out. Now, the other thing to remember here is that glucose is being oxidized to pyruvate. How do we know? Because NAD plus is being reduced to NADH, and you can't do a reduction without an oxidation. So where did the electrons come from for reducing NAD plus to NADH? They must have come from glucose, because that's the chemical that went in. What is the chemical that comes out? Pyruvate. So glucose is oxidized to pyruvate, and along the way, that allows NAD plus to be reduced to NADH. So already we're starting to see redox reactions. And we're starting to see the end result of those redox reactions. Now, if you're an organism that functions in the absence of oxygen, this may be the only way that you can make ATP. Because if you lack a mitochondrion, which is the case in anaerobic bacteria, or you don't have any alternative way of making ATP other than glycolysis for a number of different reasons, this is your only way of making ATP. It doesn't make much, 2 net yield of ATP. But what we have to bear in mind is even a small amount of extra ATP is better than nothing at all. Because if you have no extra ATP, you're out of luck, you're dead. So the small amount of ATP that's coming from glycolysis, big deal for those organisms that are considered to be anaerobic. Because this is it, this is their ATP. They're not going to be very chemically active. They're not going to grow very quickly. They're not going to reproduce very quickly. But they have enough energy to keep themselves alive because they've always got a way to make or to regenerate the ATP that they're constantly burning up. They can get it back. So this is a big deal for them. But for those organisms that are dependent on large amounts of ATP, need to generate large amounts of ATP from the energy of their food molecules, this is just not going to cut it. It's not enough energy, it's not enough ATP. So we need to be able to take advantage of that. So the question becomes, if we're getting energy out of glucose, where is the vast majority of the energy in the glucose if very little of it is released as ATP here? Well, we know some of it's in the NADH, but even that's only a little. So where is the vast majority of it? Well, that's in the pyruvate. So if we want to continue to extract energy out of glucose, we need to process the pyruvate. So let me just add a note here. So we need to remember that most of the energy here is still locked away in that pyruvate. So we've got to process the pyruvate. So in glycolysis, just a quick concept check. We're not going to stop here for very long at all. In glycolysis, which molecule is oxidized, which is reduced? Well, I just gave you the answer to that one, didn't I? The molecule that is oxidized is glucose. The molecule that is reduced is NAD+. So glucose is oxidized to pyruvate. Might as well put that in here, glucose. And NAD+ is reduced to NADH. So that's the first step of cell respiration, glycolysis. The only part of cell respiration that functions in the absence of oxygen, and the only part of cell respiration that functions in the cytoplasm or the cytosol. From here on, we're going into the mitochondria. So the first step is to get the pyruvate, that's the end product of glycolysis, into the mitochondria, which means it's got to cross the mitochondrial membrane. And the way that it gets across is via facilitated diffusion. So there's actually a carrier protein that allows this to happen. And facilitated diffusion makes sense here because we need to bear in mind that the pyruvate is constantly being churned out in the cytoplasm, but it's constantly being consumed inside the mitochondria. So there's always going to be a relatively high concentration in the cytoplasm and a relatively low concentration in the mitochondria. So facilitated diffusion makes sense because you've got a gradient, but the molecule is too big and it is charged. It's the pyruvate ion. It's the pyruvate ion of pyruvic acid. So as an ion with a charge on it, it's not getting across the membrane without some help. And so facilitated diffusion makes perfect sense here. And so there is a carrier protein that specializes in moving the pyruvate into the mitochondria. Once it gets into the mitochondria through the transport protein, then rather than the pyruvate entering directly into the citric acid cycle, which is the next major step of cell respiration, it's going to be groomed, and the grooming results in the loss of a carbon in the form of carbon dioxide. So pyruvate, and one of the easiest way to keep track of what's going on here in cell respiration is to keep track of the number of carbons that are involved. So we started out with glucose having 6 carbons. We've cut that in half, and we've now got pyruvate, which is a 3-carbon molecule, and we've got 2 of those. So that accounts for the 6 carbons from glucose in the 2 pyruvates. Even though we're only showing one of the pyruvates here, there are 2 pyruvates. So let me move this box out of the way for a minute. So here you can see the pyruvate molecule, and here's the acid group. And the acid group's the key here to understanding what's going to happen next. Because the acid group is a carbon plus 2 oxygens. That's also what carbon dioxide is. So the loss of this acid group is what results in the carbon dioxide. What's left is a 2-carbon group, which is referred to as an acetyl group or acetyl group. It's relatively unstable. And so to stabilize it, a coenzyme molecule called coenzyme A is temporarily attached to it to provide a stable molecule that can then be utilized in the citric acid cycle. It's a way of moving it to the citric acid cycle. But in the process of losing this carbon dioxide, in the process of losing the carboxyl group, so maybe I'll put this in here. So this is a carboxyl group. That's what's being lost as the carbon, well, that's what is lost to give rise to the carbon dioxide. So in the process of losing that carboxyl group, a pair of electrons is released or is made available that can be used to reduce NAD plus to NADH. So here is another source of NADH. And again, remember there are 2 pyruvates that are coming through. So that means we get 2 NADHs here. We get 2 acetyl-CoAs here, but it also means we get 2 carbon dioxides being released. So it's the acetyl group, and it's not actually the acetyl-CoA, but it's the acetyl group that's going to enter the citric acid cycle. Now, why should the acetyl group enter the citric acid cycle? Well, I can show you a really complicated diagram. And this is the really complicated diagram. This is not the full citric acid cycle in all of its details, because even though it's got the names of the intermediate molecules and it's got their structures in there so you can see what's going on, doesn't include all the enzymes. Every step has an enzyme involved. And for those of you who are planning to go on and get a biology degree and are going to be taking some advanced biology classes, so this doesn't mean anyone who's going to the health sciences or the nursing program. You escape learning a whole lot more about the citric acid cycle. But for those of you who are planning to go on to medical school or vet school or something like that, pharmacy school, you will, in your more advanced biology classes, learn this in intimate detail. Names of enzymes, names of molecules, structures, the whole 9 yards, because it becomes that important. So sitting at the top here, we have the acetyl-CoA group. Remember, it's got 2 carbons in it. As we follow around the diagram, notice that 2 carbon dioxides come out. Well, that's the loss of 2 carbons, which is the equivalent of the 2 carbons in the acetyl group. And if this is a cycle, and it is, whatever we take out, we have to replace. So if we're going to take out 2 carbon dioxides, and we will, we have to replace the carbons of those 2 carbon dioxides with 2 carbons from the acetyl group. So that's why it's the acetyl group with its 2 carbons that comes in to replace the 2 carbons that are lost as part of the cycle so that the cycle can continue. Along the way, again, every time we lose a carbon dioxide, we make available a pair of electrons that can be used to reduce NAD plus to NADH. So we produce some more NADHs. There is another way of getting electrons by taking hydrogens. Remember, a hydrogen, we can put in here, H equals H plus an electron. So when we add 2 hydrogens, we're actually adding 2 electrons, and those electrons, it turns out, are useful. They can be, or they do contribute to the generation of ATP, as you'll see. There is another way of extracting electrons through a slightly different form of chemical reaction that also makes electrons available for reducing NAD plus to NADH. So in total, for each turn of the citric acid cycle, we make 3 NADHs plus an FADH2, which itself contains a pair of electrons that can be utilized. Then also notice that not directly, but indirectly, as a product of transferring a phosphate around, we can generate an ATP for every turn of the cycle. So the important thing to remember here is this is per acetyl-CoA. But remember, we get 2 acetyl-CoAs per glucose molecule. Each pyruvate, we get 2 of those from a glucose molecule, gives an acetyl-CoA, so we get 2 acetyl-CoAs, which means per glucose, the cycle goes around twice. Now, before you get all panicky and start wondering, so why do I continue talking about this if I'm saying that you don't need to know this? It's just so that you understand that there is a background to this next diagram. There is an underpinning to this diagram, because this is what I really want you to know. Okay, so remember again, what I'm emphasizing here is chemically what goes in, chemically what comes out, energetically what goes in, energetically what comes out. So if we're thinking in terms of what goes on related to the citric acid cycle, we start with the pyruvate. And for each pyruvate molecule, we lose a carbon dioxide as it gets groomed. And this step is actually referred to as so why should it be called pyruvate oxidation? Well, remember, as part of the loss of the carbon dioxide, a pair of electrons are used to reduce NAD plus to NADH. Again, electrons, energy, can neither be created nor destroyed. They don't magically appear out of nowhere. They've got to come from somewhere. And if you're going to do a reduction, you've got to do an oxidation. You can't have one without the other. And so the only source of electrons for the reduction is the pyruvate itself. So the pyruvate is what is giving up the electrons. Therefore, the pyruvate is being oxidized to the acetyl group. The electrons are then being used to reduce NAD plus to NADH. So there's your reduction. We know that that's a reduction, so something else has to be oxidized. What's being oxidized here is the pyruvate, and the pyruvate is oxidized to the acetyl group, which is then stabilized by the CoA. So for each pyruvate that enters the mitochondria, we lose a carbon dioxide, but there are 2 pyruvates per glucose, so there are 2 carbon dioxides released here. That produces 2 NADHs in this step. So we need to remember that. Then as we go around, we need to remember that if we're thinking in terms of glucose, we have to double down on everything. Because, let me update this one and do it the same way as I've done it down here. Because again, we have to remember that we get 2 pyruvates per glucose molecule. So the numbers here are correct if we're thinking single pyruvate molecule. If we're thinking per glucose, then we have to double down on all of these numbers. So at the end of the citric acid cycle, we have finished the chemical disassembly of glucose, and all of the glucose, or specifically the carbons from the glucose, are now in the form of carbon dioxide. So if we do the math, we can see that. So we look at the carbon dioxide coming from the pyruvate oxidation. There's a single carbon dioxide there, plus we've got 2 carbon dioxides coming out of the So these 3 carbon dioxides, the one from pyruvate oxidation and the 2 from the citric acid cycle, represent the 3 carbons that came from the pyruvate. But again, if we're thinking in terms of the glucose, it's 2 pyruvates that come from the glucose. So instead of just 3, we now multiply by 2, and that would become 6. And there are the 6 carbon dioxides that are the end product of the breakdown of glucose. So at this point, we have chemically disassembled the glucose. So that's where the energy is. Now that we've finished the chemical disassembly of the glucose, the energy is in the form of those electrons in the NADHs and the FADH2s. So obviously, in the last step, the 3rd step of cell respiration, we're going to be looking at how we get the energy out of those electrons to make ATP. But before we go there, a quick concept check again. Again, not going to stop the recording. We're going to just stop for a concept check, make sure that we've got everything squared away here before we move onwards. So at the end of the citric acid cycle, glucose has been fully chemically disassembled. Where is the energy that's been extracted from the glucose stored at this point? I think I just answered that one, but yeah. Which process in your cells are responsible for producing the CO2 you exhale? I think I might have just answered that one too. So where is the energy from the glucose? In this case, it's in the electrons. Let's tidy that up a little. And what are the processes that produce the CO2? Pyruvate oxidation. One per pyruvate. And the citric acid cycle. And that's 2 per pyruvate. So if you thought along those lines, if that was where your thinking was running, good, because you're starting to put the pieces together. Anyway, onwards and upwards, let's talk about how we get the energy out of those electrons. And no real guesses as to where we're going here. I've introduced you to the idea of an electron transport chain. That's where we're going now. So the umbrella process here is called oxidative phosphorylation. Oxidative because we're oxidizing NADH. We give the electrons from NADH and FADH2 are being processed. So they are being oxidized, and the energy that's being released from those electrons is then going to be used to generate ATP, a process that is called a phosphorylation process. Anytime you add phosphorus to something, it's called phosphorylation. So we are phosphorylating ADP to ATP. So that's what we're going to be doing. The energy, however, is not used directly from the electrons. Instead, we create a hydrogen ion gradient. And as we've seen before, gradients can represent a source of energy. And a flow of hydrogen ions represents a very serious source of energy for cells. We refer to it as a proton motive force. Hopefully you remember that again from Chapter 7, when we talked about movement across membranes. We talked about the proton motive force. And so here we're going to see, again, a utilization of the proton motive force, this hydrogen ion gradient, as a source of energy for generating ATP. That's the direct source of generating ATP, or the energy for generating ATP. It's the hydrogen ion gradient. So somehow we've got to turn the energy of the electrons, their free energy, their potential energy, into a hydrogen ion gradient. And that's what the electron transport chain does. So here is a diagram that illustrates the electron transport chain. And we can't see the top of it because of this box. So let me see if I can move this. We'll move it to the bottom for the time being. You can temporarily sit down there. So notice that the vertical axis here is free energy, with 0 being at the bottom and 50 being at the top. I don't know whether those numbers are accurate. It doesn't really matter. We go from low free energy at the bottom to high free energy at the top. And down here at the very bottom is oxygen with very low free energy. Now, I've already told you that there is a connection between free energy and electronegativity. We know that oxygen is extremely electronegative. It's the second most electronegative element. And that's why it has low free energy. Because remember again, it's the electrons that represent the source of free energy, or the potential energy that can be released as free energy. And given that oxygen holds on to its electrons very tightly, it is electronegative. It doesn't give up those electrons willingly, therefore it doesn't have a lot of free energy to give away, essentially none. At the top, we have NADH, which is weakly electronegative, meaning it will gladly give up its electrons under the right circumstances. So consequently, because it has electrons to give away, it has free energy to give away. So it has high free energy, low electronegativity. And that's really all you need to know to understand how the electron transport chain works. We are moving from NADH with weak electronegativity, but high potential energy, sequentially to more and more electronegative and therefore lower and lower free energy situations or protein complexes. And so as you move from a region of, or a molecule that has high free energy and low electronegativity, into sequentially more electronegative molecules and therefore sequentially less free energy, there's got to be a loss of energy. And so as the electrons, which are the things that are moving through this electron transport chain, progressively move to a more and more electronegative environment, representing a lower and lower free energy environment, ultimately winding up in the ultimate low free energy, high electronegativity situation, oxygen, there is a surrender of energy. So the question then becomes, how can that loss of energy give rise to a hydrogen ion gradient? Well, the answer is not terribly complicated. Remember that nature does not like unequal electrical charges. We've seen that. A hydrogen atom equals a hydrogen ion plus an electron. And the reason why that hydrogen ion is always present is to maintain electrical neutrality. So when the electrons start making their way through this progressively more electronegative, lower potential energy pathway, they get chased by hydrogen ions. And each NADH and each FADH2 give up a pair of electrons to the electron transport chain. So consequently, each pair of electrons has to be chased by a pair of hydrogen ions. So when the electrons move from NADH into the first of the three main chemical complexes that make up the electron transport chain, they are followed by a pair of hydrogen ions. So let me bring this up again. So a pair of hydrogen ions enter as the electrons into the complex and follow the electrons through the complex. When the electrons eventually enter the first of two mobile proteins that connect these complexes together, called Q. Q carries the electrons away from the complex. Therefore, these hydrogen ions don't have to follow. And so those hydrogen ions move out of the complex. But rather than being returned to the matrix from which they came, they go into the intermembrane space. So effectively, these hydrogen ions have been moved across the Christie membrane because this complex of proteins is sitting in the inner membrane or the Christie membrane. So when the the hydrogen ions are picked up from the matrix to chase the electrons through complex number one, when they're released, rather than coming back to the matrix, they're released into the intermembrane space. They've been moved across the membrane. Q carries those electrons into the second complex, which is called complex 3. We'll talk more about complex 2 in a moment. I haven't forgotten it, it's there. But complex 3 is the next mainstream complex. And again, another pair of hydrogen ions enter in, again coming from the matrix to chase the electrons through complex number 3. When the electrons then enter into the second mobile protein called cytochrome C, those hydrogen ions are no longer necessary. And so they enter into the intermembrane space. They're not released back into the matrix, they're released into the intermembrane space. And so you have another pair of hydrogen ions entering the intermembrane space. The electrons now leave cytochrome C and enter complex number 4. And you can see what the process is going to be here again. The electrons are chased by another pair of hydrogen ions, again, coming from the matrix. And when the electrons finally exit complex number 4, to be combined with hydrogen ions and oxygen to give rise to water, again, that pair of hydrogen ions are no longer necessary, and they are released not back into the matrix, but into the intermembrane space. And so as the electrons pass through the three complexes of the electron transport chain, moving from a high free energy, low electronegativity to a low free energy, high electronegativity environment, energy is lost in the form of the hydrogen ions that are moved effectively across the membrane when they are used to chase the negative charge of the electrons through the three complexes of the electron transport chain. But rather than being returned back to the matrix from which they came, they're returned back, or they're released into the intermembrane space, building up a concentration of hydrogen ions. So that's the electrons from NADH. What about the electrons that are in FADH2? Well, notice that FADH2 is a little bit lower on the free energy scale. And that means that the proteins of complex 1 are unable to extract the electrons out of FADH2. They can extract electrons out of NADH because they're way more electronegative than NADH, but they're only moderately more electronegative than FADH2. So there is another complex, and we believe that it actually contains the same iron sulfur protein that's part of complex 1, but it's in a different chemical environment that means that it can actually extract electrons out of FADH2. And rather than passing them into complex 1, the electrons enter Q and then enter complex number 3, and then follow the same pathway as the electrons from NADH. But what that means is they don't represent the same amount of energy. Let me put this up so I can pick up a different color. Let's use blue this time. So for FADH2, the hydrogen ions again come out of the matrix and into the intermembrane space, but they only do so through complex number 2, or sorry, complex number 3 and complex number 4. So for every FADH2, it's 4 hydrogen ions that are moved. For NADH, it's 6 hydrogen ions that are moved because the electrons from NADH pass through all 3 of the mainstream complexes, whereas the electrons from FADH2 only pass through complexes 3 and 4 and move hydrogen, and their energy used to move hydrogen ions. So FADH2 electrons only move 4 hydrogen ions, NADH electrons move 6 hydrogen ions. What that means is that the NADH electrons make a slightly greater contribution to the overall hydrogen ion gradient than the electrons from FADH2, and that enters into our energy budgeting for the production of ATP. The contribution of electrons from FADH2 to making ATP is a little bit lower than the contribution of electrons from NADH. And we have to factor that into our consideration in terms of the overall ATP production due to cellular respiration, as you'll see in a moment. So the final piece of the puzzle is, what do we do with those hydrogen ions? Well, they come back through a carrier protein complex that's connected to an enzyme complex that actually makes the ATP. So as the hydrogen ions come back through the carrier protein, so let me label this carrier protein complex. The flow of hydrogen ions causes that carrier protein complex to rotate, which in turn rotates a connected rod of protein inside the enzyme complex. And the turning of that rod inside the enzyme complex is what translates the energy of the flow of hydrogen ions into mechanical energy that forces the enzyme complex to open and close to accept ADP and a phosphate group, and then close around those to trigger the production of ATP. So it's that mechanical effort or that mechanical work being done by the rotating rod that is rotating in as a result of the flow of hydrogen ions that represents the actual energy for forcing the enzyme complex to open and close to accept ADP and PI as the substrates and bringing them together to promote the formation of ATP. And your cells, every cell, well, every mitochondrion in every cell, and there's usually about 30 to 40 mitochondria per cell, contains thousands of these little ATP complexes called an ATP synthase. So this whole complex is referred to as ATP synthase. So it's an enzyme, it ends in ASE, so we know it's an enzyme. And this is the enzyme complex that carries this out. And you've got thousands upon thousands of these in every mitochondrion, in every cell in your body. And it's the ability of this complex to tap into the energy of the flowing hydrogen ions and turn that into mechanical energy that allows us to make ATP. And not surprisingly, figuring this out has earned a number of Nobel Prizes. There's been about, last count was around 4 or 5 Nobel Prizes since 1960 through to around 2012, 2013, I think was when the last one was given away. In some cases to single individuals, in other cases to multiple individuals. But that should come as no surprise because this is fundamental to understanding how cells process energy. This is where the energy to keep us alive comes from. So that's how you tap in to the energy of glucose, or more precisely, the electrons in glucose as a source of energy to drive the synthesis of ATP. You carry out a series of redox reactions that allows you to capture electrons in molecules that are then able to donate those electrons to the cellular staircase, the electron transport chain, a stair step of proteins that allow energy to be extracted from the electrons as they move from high potential energy to low potential energy, from low electronegativity to extremely high electronegativity. And some of that energy is then utilized to transfer hydrogen ions from the matrix to the intermembrane space that creates the hydrogen ion gradient that provides the flow of energy to drive the little machine called an ATP synthase that makes ATP. And it really is a machine. I mean, if you're turning a flow of hydrogen ions into mechanical energy that carries out mechanical work of making ATP, that makes it a machine. And so you have this flow of hydrogen ions driving these little enzymatic machines that generate all the ATP that we are dependent upon. So that's how we put it all together. That's it. So when we tie everything together and we think in terms of oxidative phosphorylation, so remember again, the oxidation refers to the fact that we are oxidizing NADH. It's surrendering its electrons to the electron transport pathway that is then generating the hydrogen ion gradient that provides the energy to carry out the phosphorylation step, the addition of phosphate to ADP to make ATP. So the electron transport pathway is or chain is the way we tap into the energy of those electrons to move the hydrogen ions, and then we tap into the flow of hydrogen ions to drive the phosphorylation step in chemiosmosis. So it's the combined electron transport chain and chemiosmosis steps that give rise to oxidative phosphorylation. So this is not a bad summary diagram of oxidative phosphorylation. Do you need to know this diagram of the ATP synthase? No, I'm not going to ask you about that. Do you need to know this diagram in detail? A little bit. So in terms of how it functions, the fact that the electrons are moving from high free energy to low free energy, from low electronegativity to high electronegativity, that it's a sequence of proteins, that you do need to know. But the names of the proteins, the sequence of the proteins, not so important. In fact, not really important at all, to be quite honest. So not surprisingly, there are a number of chemicals that interfere with the flow of electrons in the electron transport chain and therefore act as poisons. So rotenone is a very common poison for getting rid of insects and getting rid of rodents. It's a rodenticide and an insecticide, and it blocks the flow of electrons out of complex 1, which backs everything up at that point. Cyanide and carbon monoxide do a double whammy on us. They can bind to the iron in hemoglobin, which is problematic because it blocks the transport of oxygen. But they can also interfere with the exit of electrons out of complex number 4 in the electron transport chain to oxygen to form water, which causes the whole electron transport chain to back up and means that no longer can electrons flow through it, no hydrogen ion gradient, no ATP. And they do that very, very quickly and very efficiently, which is the reason why cyanide kills so rapidly, because it's interfering in 2 very important pathways. Then an antibiotic called oligomycin interferes with bacterial ATP synthase. And the good news here is the bacterial ATP synthase is different from our own. And so in terms of interfering with ATP synthesis in cellular respiration in eukaryotes, doesn't have an effect, but it does target back the aerobic bacteria. There is a little bit of a difference. And so this antibiotic can kill bacteria, particularly aerobic bacteria, by shutting down their ability to make ATP. And then DNP, otherwise known as dinitrophenol. I think I better write that a little bit smaller. Dinitrophenol is referred to as, I'll do it all lowercase. Is referred to as a hydrogen ion gradient uncoupler. And that's a big name, but what it basically means is that it supplies or provides an alternative pathway for the hydrogen ions to get across the membrane. And because that is a path of least resistance, they preferentially will flow through the DNP rather than flow through the ATP synthase. Like most things in life, people included, myself included, if there's a hard way and an easy way, our general tendency is to find just to try and find the easy way, right? Path of least resistance. And in this case, following the path of least resistance means no ATP, and it winds up killing you. What is ironic here is that DNP has been sold in the past as a diet drug, because in small quantities, its ability to interfere with the production of ATP means that you less effectively extract calorie energy out of food, which means that you will actually lose weight. The problem with this, of course, is that human nature kicks in. And if a small amount of DNP works great as a weight loss drug, then a whole lot more should work even better, right? What could go wrong? Well, if you take too much, if you go beyond the recommendation, it actually shuts down the production of ATP completely and you die. Not exactly what you were expecting. So DNP is not generally available for sale as a diet drug, and nor should you fall for the idea that it ought to be used as a diet drug. It is very, very, very dangerous because of its ability to interfere with the production of ATP. So when we want to look at the overall production of ATP from cell respiration, we can come back and we can take a look at this diagram. And you can see that in some circumstances, the amount of ATP being produced is fixed. So the amount of ATP that comes from the glycolysis, that's fixed because of the chemical reactions. The amount of ATP coming from the citric acid cycle, that's also fixed because it's the product of chemical reactions. So why the flexibility in terms of oxidative phosphorylation? Well, remember that we were producing NADH in glycolysis, and that's out in the cytoplasm. And I did mention that those electrons can enter into the mitochondria. Well, it turns out that there are two different pathways for the electrons, not the NADH, just the electrons, to enter into the mitochondria. And it depends on which way those electrons enter the mitochondria as to what the overall energy production is. Because in some muscle, the electrons pass from an NADH in the cytoplasm to an NADH in the mitochondria. And so energy equivalence is maintained. And you get the full benefit of the electrons out of NADH. But in other types of muscle, the electrons pass from NADH into FADH2. And remember, the electrons in FADH2 are quote unquote lower energy. They don't contribute as much to the hydrogen ion gradient, so they generate less ATP. So in that muscle, the overall production of ATP has to be downgraded a little bit because the electrons are coming from FADH2, not from NADH. There is a loss of a certain amount of available energy. Hence the flexibility or the variability in the overall production of ATP. So you've got to be a little bit careful about doing your math. You also need to be a little bit careful as you do research outside of the textbook. You may find to 36 to 38 ATP. And I know in the crash course video, and if I remember correctly, also in the Khan Academy videos, they talk about 36 to ATPs being the product of aerobic respiration. This is no longer considered valid. There's been a reevaluation as to how many hydrogen ions it takes to make an ATP. And we now feel that it actually takes more hydrogen ions to generate an ATP than we thought previously. So we've had to downgrade the amount of ATP being produced as a product of oxidative phosphorylation. So again, if you see the numbers 36 to 38, those are old-fashioned numbers. They're not considered to be acceptable anymore because they're based on faulty hydrogen ion mathematics. Just keep that in mind. So we are going to take a fully fledged break at this time. I'm going to stop this presentation after I give you the concept check, and I will answer these questions with the next presentation in the sequence. So here are the questions. What effect would the absence of oxygen have on the function of the electron transport chain? The function of the electron transport chain relies on the fluidity of the Christie membrane. What feature is evidence for that? I'll see you at the next presentation with the answers.